Hi everyone. In class, you guys talked about different applications of DNA technology, and we're going to be focusing in on one specific one, which is DNA profiling. So, we can actually use everything we've learned about DNA technology to help solve crime mysteries. There's a whole field of study about this called forensics, and this is when the scientific analysis of evidence for crime scene investigations. And so a lot of times it involves studying dead bodies and understanding the cause of death. So one example of this is DNA profiling, and this is when you analyze DNA samples to determine whether they came from the same individual. And the reason why this would be important is if you don't know who there was a murder and then you don't know who killed the victim, and so you'd want to analyze the DNA samples of everything in the crime scene to figure out what the DNA samples were of the actual perpetrator. And so usually for DNA profiling, when you try to figure out who is who and match based on DNA, which person was the perpetrator, you're going to look at very specific segments of DNA called DNA markers. So you're not going to just look at all of their DNA because there's just too much, but there's specific segments of certain locations of DNA that tend to be very different for every individual. And so you can kind of think of it as your own unique DNA fingerprint. So in DNA profiling, what happens is DNA samples are taken from the crime scene, and so this will be a mix of the victim's own DNA and usually the DNA of the suspect. And so if they leave behind any hair, like the hair follicles or the part where your hair touches your scalp, might have some DNA you can use. If there's any blood, if there's any saliva, any of those kinds of things would allow you to be able to determine what the DNA is of the suspects. And so you'll take all of this DNA that's available and you will amplify the DNA so that it's easier to see. So you'll take the DNA and make many, many copies of that DNA. And then you will compare the amplified DNA of the crime scene with all the different suspects and try to figure out which one matches. So for example, let's say this was the DNA isolated from the crime scene and you know it's the suspect, not the victim. You can compare it to a bunch of people you think may possibly be the suspects, and whatever matches up, you realize that must be the same person. Okay, so the question is, how do we amplify DNA? And remember, the reason why we want to amplify DNA is because here, to get these really nice, dark, bold lines, to be able to see it, we have to have a lot of DNA. So how do we do this? How do we get a lot of copies of DNA quickly? We use something called polymerase chain reaction, PCR, and this is just a technique that makes multiple copies of DNA very quickly. The way it works, we're going to use primers, which are just short little segments of DNA that are complementary to each end of the target sequence. So if we want to amplify this one section of DNA, the primers will be specially designed to just be on the very ends, so it will only code for whatever we want. So you can kind of think of these primers as very similar to the primers we saw in DNA replication. Remember, we always need those primers for you to replicate anything because DNA polymerase, that enzyme, cannot add nucleotides unless there's a primer there. So here's a picture. What happens is you have your original DNA strand, and this is what you want to amplify or replicate. You're going to heat the DNA so that you break all those hydrogen bonds. And by breaking those hydrogen bonds, you can separate the DNA. And then after that, you're going to slightly cool the DNA when you add primers. And so that will allow the primers now to attach to the locations that they need to. And notice one primer is for one strand, and then the other primer attaches to the other strand. And remember, DNA polymerase can only be built from the 5' to 3' direction. So notice that the primers will be in locations where DNA polymerase can just add right onto the 3' end. And so after heating and then cooling the DNA so the primers are attached, you're going to add DNA polymerase, which will continue to add complementary base pairs so that you can create 2 different strands of DNA. And notice that this DNA polymerase has to be a very special kind of polymerase that can withstand high temperatures. So even though we've slightly cooled the DNA so that the primers can attach, it's still relatively hot, much hotter than our normal body temperatures. And so you need a special polymerase, which they take from bacteria that grows in high temperature conditions. So as you can see, this is right here is the end of cycle 1. So cycle 1 ends with 2 new strands of DNA. This right here is when you do the same process. You heat the DNA, separates the 2 strands, you add the primers after cooling, and then you allow DNA polymerase to create new strands. So this right here would be the end of cycle 2. So cycle 1, you end with 2 DNAs. Cycle 2, you end with 4 DNAs. And then cycle 3, you do the same process and you end with 8 DNAs. And you can see that it's an exponential process, 2 to the n power for every cycles. And so you can imagine that exponentially the DNA can be amplified very, very quickly. So this is just written out version of what we discussed. We always heat the mixtures to separate the strands. We cool the strands and add primers so that we have kind of a starting point for our DNA polymerase. Then we add a specialized heat-stabilized DNA polymerase, which will add to the 5' to 3' direction, and we synthesize our new DNA strand. And this leads to just a lot of DNA sequences. Okay, so after we've created a lot of copies of our DNA, remember now what we need to do is we need to separate these different sequences of DNA so we can kind of analyze who is who. So what we do, we're going to use this technique called gel electrophoresis. This is a method that separates any kind of macromolecules. You can use it for mostly proteins or DNA, and you separate it based on size, charge, or some other property. So it's just a tool to separate our DNA fragments. And this gel has a lot of tiny little threads. Even though it's a gel, it's kind of almost like a web of different material, so that it kind of has these little holes so that bigger pieces can't go through very easily, but the smaller pieces can fall right through the holes and go right to the bottom. And so this is just a picture of what gel electrophoresis looks like. Remember, the context of this is when you're taking crime scene data for DNA profiling, you want to be able to figure out who is who by looking at their DNA sequences. And remember, if you just look at DNA and it's all stuck together, everyone's DNA is going to look exactly the same. And so the only way we can determine who is by cutting up their DNA and then separating those different fragments and seeing if the fragments are the same size. So here, the process of DNA electrophoresis. So this is gel electrophoresis just for DNA. The first thing we do is we take the DNA and we cut it into fragments. So we use one restriction enzyme and we cut it all into fragments. After we do that, we coat the DNA fragments with a chemical called SDS, and this just makes everything in the DNA evenly negatively charged. The reason why we want all the charge to be even around the DNA is because we're going to place this DNA in a gel and we're going to power electricity through electrical current through the gel. And so the top of the gel will be negative and the bottom of the gel is positive. So because the DNA is coated with this negative substance, it's going to migrate towards the bottom of the gel. And so it's kind of controlling for charge because we only want to separate the DNA based on its size. The larger fragments that are bigger, they can't fit through those little holes in the gel, so they'll tend to stay at the top, whereas the small fragments will sink right to the bottom. And so the separation is purely based on size because all of the charges are kind of equalized. After we separate the DNA fragments, we can compare samples. So if we take the same restriction enzyme and we cut the crime scene DNA and we cut the suspect DNA, if it's cut by the same restriction enzyme and it's the same person, their fragments should be the same. If it's a different person, their fragments are going to be different. Afterwards, we'll stain the DNA fragments to be able to visualize these DNA bands. And so this is a picture of a real-life gel electrophoresis. You have these different columns, and you can put a bunch at the same time. So for the person to be the same, all of the bands have to really match up. So you'll notice some of the bands look like it's about the same, like all these have the same band, but you'll notice the bottom parts are very different for each one. Okay, now the question is, we've talked about how to amplify a lot of the DNA so we can see the different fragments. Then we talked about how we're going to separate those fragments so we can figure out who is who. Now the question is, which fragments are we going to choose? Which parts of the DNA or the DNA markers are we going to use for DNA profiling? There's something called repetitive DNA. This is nucleotide sequences that are present in multiple copies in the genome. So they usually don't really have a function. They don't really code for any kind of protein, but they're just repeating sequences of like AT or GGCC, and you'll see this being repeated over and over again. There's something called short tandem repeats, and these are short sequences of this kind of repetitive DNA that can be used for genetic DNA profiles. So what happens is generally in certain locations in our chromosomes, we all have this kind of repetitive DNA, these short tandem repeats, but the number of repeats we have is different. So I might have only 4 ATs, whereas someone else might have 10 of them or 15 ATs. So like we said, the number of repeats can vary between individuals. And so we're going to do, in DNA profiling, they do STR analysis or short tandem repeat analysis, where the DNA markers they'll choose are these specific segments of DNA in our chromosomes where there's just repeated sequences of nucleotides. And these sequences tend to be very different between people. Okay, thank you guys so much for watching. Today we talked about specific techniques we can use, different DNA technology techniques with the specific application for DNA profiling. Okay, we'll see you later.